Hello, everyone, and welcome. Today, we are going to talk about GWAS, which is a very serious topic and it is the latest of the latest in the nephrology world. So, first of all, GWAS stands for Genome Wide Association Study. It is a large scale approach that compares the genomes of many affected individuals to healthy controls in order to identify genetic variants that are frequent in patients. The key point to remember is that GWAS does not directly search for a single responsible gene, as in monogenic diseases. Instead, it identifies regions of the genome that are statistically associated with an increased risk of disease. So, we're not looking for a gene, we're looking for regions or zones of the genome which are statistically associated with the risk of developing a disease. So let's jump into some definitions. First of all, as I said, GWAS is a study that scans the entire genome to find associations with a disease or a trait. SMP, it is a small variation of single base of DNA frequent in the population, and it is mostly used in GWAS studies. Association, it is a variant which is more frequent in the sick than in the controls. And by risk, we mean that the variant increases or decreases the probability of having the disease without being necessarily causal alone. And by complex disease, we mean that disease is influenced by several variants plus environment. I think here we might sum up all this by saying that a GWAS study uses SNPs to look for association that might explain a risk for developing a complex disease, and especially not a monogenic disease. Excuse me. And now let's move on to some general principle. So the general principle of a GWAS follows 5 steps. First of all, we compare two groups, affected patients and unaffected controls. Second, we perform genotyping by analyzing hundreds of thousands to millions of SNPs. Third, we apply statistics to find SNPs significantly associated with the disease. Fourth, each significant SNP signals an interest in regions of the genome called locus. Fifth, we attempt biological interpretation by linking this region to a gene or a biological pathway or a mechanism. These are the steps for any general GWAS study. So let's move on to some results. So typical GWAS results include several key elements. A very low p-value indicates a statistically strong association. The odds ratio or OR measures the increase or decrease in the risk. Locus is the genome region associated with the disease. A Manhattan blot is a graph showing peaks at the associated regions, and the QQ blot is a control graph used to verify the statistical quality of the analysis. And now, what a GWAS shows? This is the big question. We must have answers for this. So a GWAS mainly reveals susceptibility factors. It helps understand why some people are at a higher risk of developing complex kidney diseases. such as chronic kidney disease, IgA nephropathy, and certain forms of FSGS or variation in estimated GFR. However, it is important to understand what a GWAS can and cannot say. For example, it can tell us that a locus is associated with a disease, that a biological pathway seems involved, and that a risk is increased or decreased in that case, and that multiple small effects add up, but it does not tell us that a variant is necessarily causal, that a patient will definitely develop the disease, and that the result is sufficient for individual diagnosis, or that the disease is monogenic. This is a huge difference. So we must keep in mind what the GWAS can say and what does not always say, especially when it comes to monogenic disease. I would repeat, we're not looking for a monogenic disease. We are just looking for association that might explain any complex disease. Let's give some examples in nephrology about GWAS. So GWAS have been used to identify some locus associated with estimated GFR and kidney function. For example, the U mode locus is associated with kidney function and the risk of chronic kidney disease. In IgA nephropathy, associations have been found with HLA and mucosal immunity and complement and IgA1 glycosylation. These just are examples, and we would not forget APOL1 variants, which are important risk factors in population of African ancestry and representing a high impact genetic factor in a specific context. And finally, we must talk about polygenic scores, which combine many small effect variants to estimate an overall risk. These are some examples about using GWAS studies. Here are some essential keys you need to remember when dealing with any GWAS study. A common variant is present in a non-negligible proportion of the population. And small effect means each SMP individually increases risk only slightly, but when it is added up to another SMP effect, we would have a cumulative effect, which means that multiple variants together can strongly modify risk. Then we can say also that polygenic means that the trait depends on many variants and not only one. Locus is a genomic region, not necessarily the exact causal gene. Linkage disequilibrium means a detected SMP might be associated because it is transmitted together with the true causal variants. Replication means results must be confirmed in another population. We must do another study to confirm our results. We would not be satisfied by only one result. And then population stratification refers to a possible bias if the compared groups do not share the same genetic ancestry. I think this is quite fair. And here we're going to move to the difference between GWAS versus monogenic disease. It is important to distinguish GWAS findings from monogenic diseases. In monogenic diseases, there is one rare variant with a strong effect, such as PKD1, COL4A5, or pathogenic U-mode mutations. These are often used for individual diagnosis, and family transmission in this case is usually clear. In contrast, GWAS deal with complex diseases involving multiple common variants with small effects such as loci associated with GFR or IgA nephropathy. And GWAS mainly serve to understand risk and mechanisms, and family transmission here is less straightforward. So these are the main differences between any monogenic disease or a GWAS complex disease. This kind of studies might be practicable and might serve to better understand the pathophysiology, including immunity, complement, glycosylation, and tubular function. They allow us to identify new biological pathways such as the U-mode pathway, HLA, and complement. They can be used to stratify risk through polygenic scores, although this is still limited in routine clinical practice, and they support also therapeutic research by identifying potential drug targets. So I think these kind of studies are so handy and practical in day-to-day nephrology practice. But it has sadly some limitations that which are important to keep in mind. For example, association does not equal causality, and an associated SMP is not necessarily the one responsible for the disease, and most variants have only small, modest effects. And results are population dependent and may vary by ancestry. And large sample sizes are needed to detect reliable associations, and in practice, GWAS results are not yet used as individual diagnostic tests in most clinical situations. And if we had one final takeaway message to memorize, is that a GWAS is a large statistical study of the genome that searches for common variants associated with disease risk. In nephrology, it mainly helps us understand complex kidney diseases, the biological pathways involved, and polygenic risk. However, it does not replace monogenic genetic diagnosis in a given patient. The 5 keywords to remember are association, meaning the comparison of patients versus controls, SMP, which is the common variant being studied, and Lucas, which is the associated genomic region, and polygenic, meaning multiple variants with small effects, and risk, which means like susceptibility rather than a definitive diagnosis. I think we we came to the end of this session. I would to say thank you for make it till here, and see you next time, guys.